leading aluminum technologists of the country, has more than 30 years of experience in the aluminum industry. He has published more than 150 research papers in various national and and also 12 national and international patents in his credit. He has authored more than 1,000 popular science articles in Korea Daily newspapers, magazines, periodicals for the last 30 years in the regarded popular public school of Odisha. He has credited more than 300 science technology videos in the YouTube. He has published 20 books in the field of science and technology, both in Korea and other English languages. He is a member of several post of professional bodies both in country and abroad. He has visited many countries of the world for higher studies and scientific technology research. He is a PhD in aluminum technology of the USA and dealing with science communication from the UK. He has received more than 1,100 national and international state awards for his outstanding contribution of science and technology and popularization of science. Some of the notables are NRDC National Technology Award 1997 Government of India, International Organization World Intellectual Property Organization WIPO, International Gold Medal 1998, Best Officer of Alpha Award 1998. In 2000 also Government of India has awarded CSIR as National Technology Award. In the year 2008, he is also the Best Scientist Writer of India in the DSC NSPC Government of India National Award of Science and Communication. Nominated as Priya Odia 2010 by NTV in 2010. Not only that, he was also first Kalina Samman Professor of Science 2010, etc. So I request Dr. Jitendra Mishra sir to deliver his talk and enlighten this scientific topic in this occasion. I request Mr. Sir to continue his delivery talk. Thank you.
Good morning, good day, good afternoon, whatever you call it. Now the technical system starts. Whatever knowledge I have, I will share with you some of my knowledges. I will give an overview. I give an overview. I am a synthetic organic chemist. Silence, please. My dear students, kindly take your seats. Silence, please. I will take 15 to 20 minutes and will give more time to Madam who will listen from Madam. I worked in Kolkata, I worked in Bangalore for my PhD and postdoctoral studies. And I had the privilege of being the students of great personality, great scientists of the country like Professor Kushi Datta, Professor B.K. Banerjee, Professor Sena Rao. In the field of chemistry, I was their direct students. Silence, please. If you kind of murmur, I cannot concentrate on my mind. My request to all of you to kindly take your seats and be silent. Be silent, please. I will give an overall view. I will not go to the tidbits of chemistry or pharmacology. I will give you an overview regarding drug discovery and drug development. How to discover a drug? Why to discover a drug? Where to discover a drug? The first question is, what is a drug? Is it required to be developed over the years? And it required a good laboratory, good setup, good infrastructure to develop the laboratory, develop the molecule, develop the formula. And for that, you need innovation. I will give you the overall view, the dreamy picture, overall view. Unless you have an innovative mind, if you are a dreamer par excellence, suppose you are not a dreamer par excellence, you cannot innovate, you cannot innovate a molecule, you cannot design a molecule, you cannot formulate a molecule. So, formulation of a molecule, new idea, new synthesis, you have to think differently than others think in the room or others in the profession. So, you have to think differently, act differently, do research differently so that new ideas evolve, new innovations evolve, new molecules evolve, new formulations evolve. So for a discovery of new molecules, you should possess a different brain structure. You should have a different mindset. You should have an innovative bent of mind. You must be an innovative person. That is why we have added that word innovation to science, technology, and innovation. The Ministry of Science and Technology, Government of India, is going to be renamed Department of Science, Technology, and Innovation. So innovation is now the key word. And to innovate new molecules, new development, new structures, new ideas, you must have an innovative mind. And development is a continuous process. Development, there is no full stop in development. It is a continuous process. Any molecule you design, any idea you germinate in your mind, it has to be updated time to time because things are changing drastically. And in the other words, India is concerned, you call it India is the pharmacy of the world. Right now, India is known as the pharmacy of the world. Many beautiful molecules, good molecules have been synthesized here in this country. And as you know, this COVID vaccine, as a result of discovery of COVID vaccine, India is known now globally. Image of India has enhanced globally due to this COVID-19. So India has a remarkable place, particularly in the field of pharmacy, pharmaceutical science and technology, and industries involved, particularly industries in Pune and Mumbai. They are doing tremendously good work developing new molecules, particularly generic medicines. Now the age of generic medicines are coming. We have our DNA is different. So DNA specific molecules are being developed now. The new development in technology is DNA specific, individual specific. Generic medicines are being developed, modernized, and day to day new laboratories are synthesizing generative medicines based on our DNA structure. Since human beings differ from DNA to DNA, that is why generic medicine have been developed and new ideas are germinating to develop new molecules specific to the particular person. That is the beauty of new development, and that is how the drug has to be discovered. The drug has to be discovered depending upon the characteristics of that particular person. Everybody, the biological map, biological skeleton, biological design of a particular man must have. Now in India, we don't have that system. In foreign countries, we have. Everybody, once he is born, his biological map is there. His biological structure is there. There is a booklet like this. It is given to him. His biological profile. Biological profile of each and every person is born. Once it is born, the biological profile is given to the parents. In our country, that system is not there. So once the biological profile, that the booklet is handed over to the parents, once the baby is born in the hospital. Entire the biological structure, biological profile, biological design is given, and what are the diseases to come in the future to the baby. Now the designer baby consult has come. You want to give a designer baby consult has come. Whatever time of baby you want a Osura eyes, Einstein's brain. So synthesis of new things are happening. You can get your baby as designed as you like. That is the beauty of the technology now. Designer baby concept. So in our country, this biological profile booklet is essentially required so that in future, whatever diseases are going to attack me are known at the time of birth. At the time of birth, you will know what are the diseases I am going to face in my future. That depends on the biological profile of a person. In future, in various ages that I want to cross in the future, I will know what are the diseases will be attacking me in my future. That you will know at the time of birth. At the time of your birth, you will know I will be a cancer patient, I will be a TB patient, I will be a this and that patient. So you will be known at the time of birth itself. So at the time of birth, your DNA profile, DNA mechanism, DNA structure will tell you what are the diseases are going to be attacking you in future. So that you plan according to your life cycle. Life cycle or lifestyle, whatever you call it, you design it, plan it so that you live a life of that in that direction. So that is the molecule, that is the new idea. New synthesis of molecules are designed for that. You know the diseases which will come in the other 5, other 10, other 20, other 50. I know the diseases that will be attacking me. So that is the biological profile. Second is environment. Even if your biological profile is okay, you know what are the diseases going to attack to you. If environment changes, everything changes. your food, environment, air, oxygen concentration, everything depends on your growth and stability. You know, gradually oxygen percentages are coming down to the air. Air is now polluted. So even if your biological profile is good, still you will attack with diseases. So your growth, stability and strength depends upon environment. So environment will play a major role. Biological environment and atmospheric environment. All these environments will play a major role in changing your biological profile in future. So that is again another important issue which will be affected in future. So we have to plan drug discovery, drug development and drug commercialization. I was telling in the morning, collaboration. You get service in various companies. There is a big signboard in your college. There's so many companies, they attract students, they give you jobs. But you do not have collaboration with them. Scientific collaboration, commercial collaboration, technical collaboration, you should have with these companies. They recruit your boys, okay. But you should have scientific exchange program. So 1 mind, 10 minds. There is a difference between 1 mind and 10 minds. So synthesis of new ideas, synthesis of new brains is essentially required now. 1 man is not sufficient. 1 brain is not sufficient. 10 brains have to be synthesized. 10 universities have to come in hand. So academia, I was telling Professor Saturday, academia, industry, and research institutes should come together for innovating new ideas, new molecules, new designs, new drugs. So in this institute, there should be collaboration. Research should be collaboration. Collaborative research. They will sponsor projects. Industrial houses will sponsor projects. They will give you money. They will sponsor projects. They will do work. There people will be guided. Here people will be guided. Both will be guided. Guide from academia, guide from industry. They will work in industry. They will work here also. So that exposure develops over the years. So nowadays, it's the age of collaboration, scientific and technological collaboration. You should work in industries, you should work in academia, you should work in laboratories also parallelly during your professional life. So that is why your exposure is less. If you work in industrial environment, if you work in academic environment, if you work in institute, laboratories, your exposure will be manifold. You are now limited to particular environments, colleges, academics, but you don't need industry. Have you seen industries? No. Have you known how the scientists are working in industry? Do you know how the drugs are being actually developed? Have you seen the equipment where the drugs are actually done? How the synthesis, how the molecule is designed, how the scientists are working? You must go to the industry, see actually what they are doing. Hands-on experiments you are not doing. So if you visit industry where actually drugs are developed, where the actual molecule is synthesized, where the actual molecule is designed, your knowledge will be enhanced manifold. So you should visit industrial houses, not that they give you a job only. You should visit industrial houses and see, interact with the faculty, interact with the scientists, interact with the worker, and see actually how it is done. And you should work there also. You should work there. There people will come here, they will work here, and you will also go there, work there. So scientific exchange program, research workers exchange program, research scientist exchange program. All facilities may not be here, all facilities may not be there. But various industrial associations, academic associations are essentially required for the growth of any research project. 1 man is insufficient for anything. That is why Japan is number 1, because they don't work unitary. They work in collaboration. Japan is number 1 in the entire world because they synthesize one brain with another. They work in cohesion, they work in collaboration, they work individually, but they work in cohesive. So you must join hands, join minds, join ideas, join brains, so that new ideas will emerge. New ideas will only emerge through 10 brains are synthesized. And that 10 brains come from industry, academia, and research institute. But nowadays, what we find, I am king in my own domain. I am a scientist, I am king in my own laboratory. I don't know what is happening. I have seen color paper, I don't know what is UNESCO. I don't bother who is UNESCO. But I have seen color paper. So you are limited, your periphery is very limited. Your periphery is right now is very limited. It should be expanded. Expanded beyond the limit. Boundaryless. Science is a boundaryless. Science has no boundary. Your students, do you have some industry exchange program right now? Sir, Tata sir, do you have a right to exchange scientific exchange program? Do your students during their study here visit industries? How many industries they visit? 1 year, one student. How many industries he visits? For 4 years, only 1 month. How many industries during that 1 month? How many industries in 1 month? One industry. 4 years, one industry, 1 month. This is sufficient? No, this is not sufficient. So my request to you, with my folded hands, the people those who are giving you jobs, you should have a scientific, technical, technological collaboration with them. They will invest money in our laboratory. Here, they will invest money. Your PhD students will work in their money. Your PhD students will work here, will work there also. They will come and teach here. Industry people will come and teach here also. You will go and guide their workers also. They are scientists also. That is how scientific exchange program. So not only job is one aspect, but the migration of brain, that is essential. So that is my suggestion to the management. That in the future, you must visit more industries, so that linkages are there. Linkages, scientific linkages, scientific cooperation, collaboration are essentially required. One brain is not sufficient. Whoever will be. Whatever knowledge you have, whatever degree, whatever exposure, one brain is not sufficient. 10 brain synthesis is essential because this is the age of collaboration. America cannot do anything without India. India cannot do anything without America. So nowadays, it's the age of collaboration, cooperation, cohesion. That is why Japan is number one. They work in a team. We work as individual. We in India work as individual. Japan don't work individual. They work as a team. So working in a team is essentially required for growth of technology and new ideas, new development of molecules. Any new idea means synthesis of new brains, innovative minds, exposure, scientific exposure is required. I have seen a current example at Dr. UNESCO. UNESCO is a global body. He is the only one who given a certificate. I have seen in Punak. You are limited. You are limited to your periphery. Within boundary, this is my life, this is my knowledge. So science is boundaryless. Science is collaboration. You must know what I don't know whether it is some. Gita no hai. Odisha परिचय से गीता से गीता खाली एक्सपोजर गीत न गीत तो असला गीत वंदे उत्कल धरणी इज द गीत व्हिच सिग्निफाइज मी मोर परिचय से गीत मु जानि तो आवर नॉलेज इज वेरी लिमिटेड गीत गावा में गीत गाइले गीत बाजेला कसा से बट आई डोंट नो व्हिच राइट मोर परिचय से गीत से जड़ित एक्सपोजर गीत नहीं किंतु गीत बोली ना तो बहुत कथा रही जानिया भाई तो नॉलेज इज वेरी लिमिटेड तो बी ए बाउंड्रीलेस मैन साइंस इज बाउंड्रीलेस नो डोंट लिमिट टू कंट्री डोंट लिमिट टू इंस्टीट्यूट ओपन द एक्सपांड ओपनिंग वन ओपन शॉप एक्सपांड आपण गमे साइंस स्टूडेंट में फिजिक्स जानि गेली सिविल में तो फिजिक्स रे पहला नंबर पर जो दाखला करना डिफरेंट है ये मस्ट नो मो पोटेंशियल पढ़ी जो संविधान कोन जानि so everything wants to know because you have to open up unless you open up a valid new idea will not germinate. Unless a new idea is germinated, new drugs cannot be developed, new molecules cannot synthesize, new design you cannot do, new thinking cannot come. So nowadays, that is why my request to all of you is boundaryless. You expose yourself, open up yourself to new people, new ideas, new institutions, new industry, so that future belongs to you. Thank you so much. Thank you.
Thankful to Dr. Sir was lively, energetic, very aspect silence service. Also, elaborate about the disease. Without meeting our questions, not about them. So, science told about the disease, its protected, and because it is protected of the other area, and it related to drug study. And the government told about aspects of the collaboration, collaboration is something not be easy. Without collaboration, understand our development. Entity to be not to collaboration and questions are asked of you. How is going to prepare research? What is the aspects? What are the parameters? How to overcome these parameters? So is science. Belongs to science. Science. And science. So one thing is science. You are all supposed to aluminium science. Someone is working on aluminium for a pharma and medical stories. Advanced in the Hano electron, advanced in the advanced system. Advanced students, advanced system. That is the program perform. So Atasi was deposited. So that is why more understanding students have to proceed in aluminum and properties. That's the book. That's the book.
Thank you, sir, for your helpful comments. Now I would like to request Dr. Praveen Kumar Sinha Mahapatra, Principal, Institute of Pharmacy and Technology, Salipur, to felicitate the speaker of the day, Dr. Chittaranjan Mishra.
Why it is Vitalium? Can everybody tell? So 100 degrees of polymer comes to the Hindalium coil and Hindalium coil is very good. Alumno salah modal to main jane dikhe, hono nai. Hindale muna khai. Hindustan alumno corporation sakti. Kale kopata muna hindal ko. Mari unit of manufacturing to hande rakhi. Hindustan alumno corporation product ki toda hindal. Kale hindale. Hindi alumno. Hindale.
Thank you, sir. Moving on, I would like to request Dr. Sunil Kumar Kanungo, Professor, Institute of Pharmacy and Technology, Salipur, to come on the stage and felicitate the Honorable Chairperson, Dr. Anjan Kumar. Thank you, sir. We have been fortunate to have such renowned personalities among us. I take immense pleasure in extending my sincere thanks to the behalf of Organizing Committee of Institute of Pharmacy and Technology, Salipur, to today's Honorable Chairperson, Dr. Anjan Kumar, and Speaker of the Day, Dr. Chittaranjan Mishra. Thank you very much for your presence. Thank you. Now it's time for another scientific session. I feel privileged to welcome Dr. Santosh Kumar Bal, Principal, Santoshi Institute of Pharmaceutical Education and Research, to kindly join us on the stage. Thank you, sir, for your presence. Moving on, I would like to request the Honorable Speaker of the scientific session, Dr. Soma Chattopadhyay, Scientist F, Infectious Disease Biology, Institute of Life Sciences, Bhubaneswar, Odisha, to kindly come and join us on the stage. Thank you, ma'am, for your kind presence. Dr. Santosh Kumar Bal, Principal, Santoshi Institute of Pharmaceutical Education and Research. He has completed his B.Pharm from Institute of Pharmacy and Technology, Salipur, in the year of 1997. He has completed his M.Pharm from Sri Jaydev College of Pharmaceutical Sciences, Naharaganda, Bhubaneswar, in the year of 2002. He has done his PhD under Berhampur University. He also has worked as an assistant professor and professor at Indira Gandhi Institute of Pharmaceutical Sciences, Bhubaneswar, from 2004 to 2016. He has more than 30 publications and has broad experience in pharmaceutical formulation and research. Now I would like to request Dr. Santosh Kumar Bal to introduce the speaker of the day to our audience.
Thank you very much. Respected Principal of IPT Salipur, Professor Dr. IPT Salipur, respected all faculty members of the dais, respected chief guests on the dais, and my dear and dear students. First of all, I am very much thankful to the team of IPT Salipur, that is the governing body members, principal sir, organizing secretary, and all team members, those who have arranged a beautiful national conference. And very important is on the very important topic, that is new approaches in drug discovery and development, opportunities and challenges. It is a very informative and very important topic for us nowadays. We have so many challenges, so many health hazards in future. So now, in my point of view, we are starting our day from the medicine and ending our day with medicine. So this conference and this topic will give so many important aspects and objects to all of us, that is important for our health sector and our professions. I am very much proud that Dr. Soma Chattopadhyay, a scientist, infectious disease biology, Institute of Life Sciences, Bhubaneswar, is present with us. Thank you, madam, for your presence. Dr. Soma Chattopadhyay is one of the most prominent molecular virologists at the Institute of Life Sciences, Bhubaneswar. She completed her BSc in Zoology from Ethno College, Kolkata, and MSc from Kolkata Science College under Kolkata University. After completing her PhD in NIST, Kolkata, followed by postdoctoral work at the University of Connecticut Health Center, USA, she joined IPT ILS in 2007 as a scientist. Dr. Chattopadhyay is working on viruses for the last 25 years. She has worked on several human viruses, including rotavirus, HSV1, chikungunya virus, JV, and SARS-CoV-2, defining the functions of the non-structural proteins during replication, identifying the cellular proteins required for the viral life cycle, and developing antivirals are the main objectives for a research group. She has several important publications, that is more than 60 numbers, in international peer-reviewed journals, authored several review articles, has filed 5 Indian patents, developed 5 products, and supervised more than 15 students for PhD and postdoctoral research work. She has conceived and implemented a large number of extramural projects supported by DPT, DST, CSIR, IBST, VIRSC, etc. Apart from her own research area, she has extensively worked at the forefront of initiating COVID-19 sample testing and research at ILS. Recently, Dr. Chattopadhyay was elected as a NASIS Fellow 2022 and also received the IBS Fellow Award 2021 from the Indian Virological Science Society in recognition of her contributions to medical virology. She is also the recipient of the NSA Reliance Industries Platinum Jubilee Award 2021 in the field of biological sciences from the National Academy of Sciences, India, and the Biotech Product and Process Development and Commercialization Awards 2019 by the Department of Biotechnology, Government of India. Thank you, madam. Your presence is all are proud of you, that your presence is highly necessary for this topic, and I think it will be a very informative and important session for us, for all the professional present here. Thank you. Please.
Now I would like to request our volunteers to give flowers to Dr. Soma Chattopadhyay.
रितिका ठीक है सुनो ठीक है
Now I would like to request our volunteers to give flowers to our honorable chairperson.
Very good afternoon to all of you. At the beginning, I would like to thank the organizer for inviting me today to this national conference, and I hope that I'll be able to guide you guys with our work that we are doing at ILS for last almost 2 decades, and being a pharmacy department and College of Pharmacy, I have picked up the work that we do on the development of antiviral drugs. And thank you for the nice introduction. I will go a little quickly because of the interest of time. But if you have interest, then we can discuss later. So, chikungunya is my field of research, but we do also work on other viruses. But today I have selected the drugs that we have developed for chikungunya or in the development process. So I will give little introduction about virus as a whole and the way antiviral drugs are developed, and then the two drugs, one which we have taken as a repurposing of the existing drug, and the one which we have synthesized. So I'll be emphasizing on this two. So you know what is virus? Basically, it is a protein code which is encoded, encoding all the viral proteins required for its life cycle. And in the center, basically, if you see the basic structure of the virus, then this is the capsid, and then the envelope proteins which are like spike, which we know now because of the COVID pandemic. And other viruses will have envelope, which you can see here, which is basically our cell membrane. The virus capsid is encoded when it comes out from the host cell. So virus is actually packaged of the genetic information. It can be RNA or DNA. So based on their genetic material, the whole life cycle is actually different. And when we talk about drug development, so based on that biology, we can actually design our drug. So this term was coined long back. The meaning is poison. So basically, virus is quite different than other pathogens. As you can see here, it's a spherical structure which is the envelope, and where we are having the envelope proteins. And what happens when it is coming in contact with any host cell during our infection? One receptor is very important, which we have learned in the last 2 years, that ACE2 receptor for SARS-CoV-2. I'm giving this example, SARS-CoV-2, a lot because now everybody is more familiar with this virus. So these receptors are very important, that the receptor where the virus will attach, and then it can enter into the host cell during infection. So once the virus is attaching, then what happens? The capsid will lyse with the endosome, and the genetic material, as you can see here, will come out. And the virus is actually behaving like a dead particle outside because they don't have all the machineries for its own replication, production of new virus particles. So what it does, once it enters into the cells, it will utilize all the host machineries for its own replication. So once it enters into our body, we know that in different viruses, there are different root. So this root is basically the presence of the receptors in different parts of our body. I'm not sure whether you guys are able to see the slide properly, but what happens that there could be droplets through which we can get infection because the receptors are in our respiratory system. It can be in the other systems which I will just explain in a minute. So based on the receptors present in different parts of our body, the virus can actually infect us either through our alimentary canal or respiratory route or through muscles or through blood contact. So these are basically presence of the receptors, which is defining that. So in case of, this is very well known for all of you, that through droplets, for influenza virus and again SARS-CoV-2, this was the root of infection, and that is why we thought that we can just cover the receptors basically with a mask, and then we can still be fine when others are infected by corona. So for other influenza virus, this is into the root, a common cold and cough. So then another one is the drinking water or the elementary system. So in case of hepatitis A virus, polio virus, rotavirus, this is wherever it is diarrhea causing pathogen, that is usually through the elementary canal. So through polluted water, we get the infection. This is another one where through the placental route from the mother, child gets infected. I am sure that all of you know about it, right? Which virus mother can transmit to the child? HIV. It's very well known, but there is another one which is rubella, like pox. In case of that also, it can cross the, it can transmit it from mother to the child. And the other one, recently the virus which is a problem in most of the Bengal as well as Orissa and other parts, that also you guys will be able to name. In recent, after monsoon, what happens? Which virus is a problem? Dengue, right? After monsoon means when the population of mosquito is more, and then mosquito will then transmit whatever the virus it has. So dengue is one of that, but it can be JV, chikungunya, and other viruses related to vector-borne viruses. The next comes another scenario where the virus can enter into our body and then just sleep. You guys will all be able to understand that. I think there are, you guys as students. Have you ever noticed that during exam you will get a blisters here? Any of you has experienced that? I am not getting any answer. Yes. What is that? I don't know in Bengali we say jot photo or something. That is herpes virus. Have you ever seen that? Right? Why during exam you get that? Because of your stress. You guys don't eat properly, read overnight, whole night reading, and the next day you will have this. Because when we are not reading, sleeping properly, eating properly, stressed, mentally stressed, our immune system goes down, the power of immune system goes down. And this virus, herpes virus, which is in our system, sleeping all the life, you will never be able to get rid of herpes virus if you are getting infection at any time. See, we all got coronavirus. I'm sure 100% of this population present here had coronavirus. If you stand up and say no, I would say it was asymptomatic. But I will not agree that you will say that I never had corona. In my experience in last 3 years, every one of us has got coronavirus infection. But today, it is not in our body. Has been cleared from our body. But whereas for this virus, if you get infection, you will never be able to get rid of that. It will be quietly sleeping, like the way seen in the picture. And it will come out or it will wake up when our immune system is down. So that is called latent infection. The viruses which are going into this latent phase is actually difficult to develop any drugs or get rid of them. Okay. So, so this is what is reactivation when the immune system is low or immunocompromised patient who are having HIV or other infection or organ transplant patient who are having immune system suppressed. At that time, the herpes virus will be coming into reactivation. And this is what I was actually telling. So at the beginning of herpes infection, you may have blisters, lot of blisters around your mouth. But after that, once there is little bit of immune suppression, 1 or 2 blisters will develop around the mouth. Now, this is something which everybody knows, HIV. In case of HIV, there are prolonged incubation periods. It can be even 10 years, 12 years. And one would not know that person is having HIV infection. And then after that, the person will start showing the symptoms. And then it may lead to death, most of the cases. But not always the virus infection is like that. Virus can also cause cancer. In case of hepatitis, we know that hepatitis B can cause cancer, hepatitis C can cause cancer. So they are cancer-causing virus. But most of the time, in viral infection like cold cough and other viral fever or other respiratory infection, diarrhea, what happens that we'll be down with fever or other symptoms for a few days or maybe a week, and then what will happen? We are all fine. I'm sure in this, season change, all of you guys will have little cold, cough, and all, right? But then we will be all fine, and we will develop immunity, will develop antibodies, which will help us to get the protection from the next round of infection. So, why do we need antiviral? When we talk about viral infection and protection from viral infection, the main thing that we are concerned is the vaccine, right? During COVID, this is how vaccine was developed overnight, because the history tells it takes 10 years, 15 years to develop a vaccine. And in the case of COVID, we got in 1 year. So vaccine is our major way of protecting ourselves from viral infection. But then do we need antivirals? And what is antivirals? Why do we need antivirals? So we still need antivirals because vaccine can be good in preventing, but viruses can be very swift, deadly, and they do mutate, right? We had a concern. The vaccine that you guys have taken. Which vaccine you guys have taken? Which vaccine you have taken during COVID? Covaxin and Covishield, right? I as much as I know, it was Bhubaneswar, which is Covaxin, Cuttack, Covishield, right? So this vaccine Covishield is developed against what? Any idea? It was developed against the spike protein. And Covaxin was the whole virus. Now, if spike protein changes, and it was changing, you all know, alpha, delta, so many viruses come, right? So, do you think that the vaccine will give the protection? It may not, right? So, then what will happen? We will make another vaccine. So we still need antiviral because the virus can change, mutate, and immediately we may need to give some remedies. So that is why antivirals are required and not that we have vaccine for all the viruses. Right? Till now. So how it is developed usually, and this is the general life cycle. So I have taken for alpha viruses. It is true for COVID also. The virus will attach to the receptor. Sorry. So the virus will attach to the receptor and then through endocytosis it will enter. Then what happens, as I was showing, that the RNA will come out and RNA will be replicated with the help of the host machinery. And once the RNA is replicated, at the same time the structural proteins are formed and again the virus particle will be formed and then it will come out taking the envelope protein from which are, it will be on the membrane of the cells, and then the virus will come. So in this whole process, as I mentioned, that apart from the viral protein, there are a lot of host proteins required in the whole life cycle. So what is happened that when we talk about antiviral, to restrict the viral replication, we can think of developing some drugs which will not allow it to bind to the receptor. Okay, or it can not allow to fuse with the endosome, or RNA replication, or the structural protein synthesis. Even if everything is happened, we can have some protein blocker, enzyme blocker, which will not allow it to come out from the cells. So these are different windows which we can think of for developing any antiviral. So generally what happens that you can screen different chemical libraries and then see whether it is actually restricting the virus, or you can have a rational approach. Like we know our viral protein, we know the function of that enzyme, and then we can develop the drug against the active site of the protein. So these are the approaches usually taken for developing antiviral drugs. So, and as I was saying that the viral protein can be targeted for it, or the host proteins which are very critical for the virus to replicate inside the cell, we can also target that. I'm not going into details because of time. Until now, we have developed several drugs. You think of drugs for herpes, acyclovir, which is very known, and then for HCV, there are interferon therapy. And for influenza also like amantadine, or maybe you guys know about Tamiflu. For swine flu virus, it was also in market, popularly used by people. And for HIV, different drugs are there, which are against developed against different infections. So today I'll talk about one drug which is telmisartan. I am sure that all of you know about this telmisartan. You know, many people take this. What is the use of telmisartan? Yes. So for against hypertensive drug, right? So that is used mostly for IPT. What we have done here is that we have repurposed this drug for our antiviral activity. And it was found that it is having antiviral activity against a few other viruses. And what we did, we were interested to see whether it can actually inhibit chikungunya virus infection. So what we did, I mean, I'm not going into that details because why we have taken, but what we did is that basically it is a HE1 blocker, and we found that viral infection can be regulated based on that. So what methodology we have used? First, we have taken this And we have checked the viral infection or maybe we have checked the cytotoxicity of this drug first to the cells, and then we infected the cells with the virus and then added the drug. And our base method is plaque assay, where we see whether the virus is still able to make the plaques. And then we see their protein expression in confocal. We see their protein also in Western blot, and we are having facts, flow cytometry, where also we can see the viral protein. And we can do RT-PCR to estimate their genome copy number. So as you can see here, I'll go pretty quickly. We can see that this is the IC50 value, and you can see in confocal microscope, this red color is actually tagging the viral protein in, and we can see with increasing concentration of the drug, we can actually reduce the viral replication inside the cell. After that, what we did, we have done several other studies to find out the mechanism, but to evaluate this into animal model, we have used C57 mice, and we have infected and then we given the drug at every day interval to the C57 mice, and then at the end of 5 days, we have sacrificed the mice, check their serum RNA level for the virus, and then also take other tissues to estimate the virus. And as you can see here, I'm not sure whether you are able to see, but what happens that the mice develop paralysis after chikungunya infection. And what we have seen here, the mice with the drug, they did not develop any paralysis or movement difficulties, and they were absolutely fine. And then we can see in the serum level, the viral RNA moves down, and also in all the tissues which we have collected, muscles, liver, spleen, everywhere we could see reduction of the viral RNA in presence of the and in the survival part, where the mice all died on day 4 after chikungunya virus infection, we can actually give 100% protection the mice to be temperatured. And you can see this is the reduction of disease score. This is with the mice which is weak, and this is with the drug. And you can see that the muscle damage that we see after chikungunya infection, we can restore it in presence of the drug. So generally, we do all these studies to evaluate the efficacy of the drug. And this is what we have done in the human PBMC. We isolated the homicide macrophage population from 3 different donors, and then we could see that there also in human PBMC, we could actually reduce the virus after addition of this tendency. So this clearly shows that the virus can be really abrogated or inhibited in presence of tendency. And this is basically our model that we think that AT1 is blocked by tendency, and this is how we can actually restrict the viral infection and inflammation and all the muscle or joint injury and organ damage, so that the mice will survive. So another story I will just quickly touch is of envisaging an IPT. This is a small molecule that we have synthesized, and we have seen the efficacy in vitro as well as in vivo. So here what happened, this is a drug, IPT, which was already in market long back for vaccinia virus, and this it was withdrawn because of some problem that was withdrawn. So what we have done, and this is high benzamidazole, which is also known for antiviral activity. So these two were conjugated, and you guys are replicating this and me. So this molecule was synthesized, and then what we did, we have looked into in silico against all the chikungunya protein. These are 4 proteins that the virus codes, and in silico we tried to see what is there in the molecular docking affinity against the virus, and we could see that in comparison to all the moieties, it has better affinity for all the viral proteins. So then we went on to check into their antiviral activity, and we found that it can actually regulate viral infection. So to find out the mechanism, we actually saw that it can bind NSP2 protein in silico. So what we in NSP2 protein was synthesized and purified, and this protein is actually having few kerosene residues. So we can actually measure their fluorescence in presence of the drug. And what we have seen that the drug is actually binding to the protein because the fluorescence is changed after adding the drug to the protein. Not going in details, and what we have found that this NSP2 protein is having protease activity, and in presence of the drug, we can actually see the protease activity goes down. So that kind of gives us the some idea that maybe through this protease activity, the virus is regulated. So again, we went on to do the eventual study, and here, like the research, we are also going to see that we can actually reduce viral infection, and this is our IC50 against the viral infection. And in the second, you can see with increasing concentration of the drug, we can actually see the less amount of viral protein, which you can see in all this quantification graph, and also for other proteins. Okay, so we established able to regulate the viral infection. And what we did here is actually wanted to understand the mechanism, how it is working, or in which phase of infection it can actually work. Because this is a new molecule, we had no idea that how it can work. So here, this is for the pre-infection and then pre-infection and post-infection. What we have done, we have added the drug before to the cells, then added the virus. And the other condition where virus and the drug were added together, the cells. And the third condition, we have virus first, then drug. So in all these conditions, we'll tell you whether it is a host factor or it is a viral protein where the drug is present. So if I just quickly summarize here, that what we have found that during post-infection, we could see that drug is inducing viral infection. Okay. And this is we have looked into the cytokine profile in culturing into raw 4.7 cells, where you can see that inflammation is a major problem during chikungunya infection, and this inflammation can be modulated after this addition of this molecule. We have done the cytotoxicity study and bioavailability study of this drug in animals, and we, according to the OECD guideline, and we could see that this is not toxic to the mice, and we could measure its Cmax and Tmax with mice experiment, and then we finally went on to experiment with C57 mice. Can I see this? I can show you this video if it works. So this mice video another place. So here actually it shows that the mice with infection cannot walk. They develop hind-leg paralysis, so the leg is sticking out. But the mice with the drug is absolutely fine, which you can see here. That the viral copy number goes down drastically. So similarly, we have seen that in serum viral RNA into all the other is reduced. And when we check the viral protein in muscles, spleen, kidney, liver, and brain, here also you see significant reduction. And if you see that, what the swelling that is developed in the mice after chikungunya infection, it is reduced significantly. And also the enlargement of the spleen is also restored. The muscle damage is also reduced. So basically, we can actually see better survivability. We could see that reduced the body weight that we see after chikungunya infection is actually restored. And this is actually this is clinical manifestation. We could see reduction after the administration of the drug. And similarly, we have looked into the human PDMs. There also we can see that in human PDMC with macrophage, monocyte macrophage population, once we do the viral infection, we can see that the virus is reduced significantly. So that clearly says that this molecule is having the potential to be a drug at some point. We are actually pursuing this one. And the last part which I want to tell you, just that we started with our director of Ajay Varila and his student, where we have taken different plant extracts and then we have done extraction with different solvents. And what we have seen that in case of chikungunya virus as well as for SARS-CoV-2, these plant extracts are having good inhibitory activity. So basically, I took 3 different examples: one from repurposing, one from the synthetic drug, and another from plant. Because of the time constant, I tried to restrict it. So there are different ways that we can actually think of developing antiviral molecules. And definitely, you know, all this work was possible because of our pharmacist, Dr. Shubudhi from Shua University. So we are biologists, but definitely a collaboration, which was, you know, talked before me, that a collaboration is very important. We understand virology, you understand pharmacy, so we can come together and then do a better work. You know, 1 + 1 is 2, but 1 + 1 in collaboration is actually 11. So we can do more than 2 when we come together and work. So with that, I like to thank Dr. Shubudhi, Dr. Parida, who gave me all the virus from PRD earlier, and Dr. Subhashi, Dr. from NICER for all the immunological work. And a lot of students are involved in this work, and especially the 2 molecule that I talked is Circus, who has actually worked on that, and all the phytopharmacy agency. And at the end, I'd like to thank you for your patience hearing, and I'll be happy if there is any question.
Thank you. Thank you very much for your very good and this topic. Very much it is a challenge virus. Now we are facing so many problems and diseases for virus. Stand very good about virus and including them receptors, and we discussed the research about the virus and all aspects and the population of the virus. Thank you very much. Even if you have any questions regarding the topic,
regarding the infrastructure regarding the what is the source of infrastructure because I know the hypertension will be at okay. So if it is something from hypertension, growth of that the drug programs and all that is not happens.
Yes. So, you know, as I said, that I am not expert in the pharmacy part, but what Dr. Shubudhi always tells that the dose that we are using is minimum, okay? And the body is having a system that if a person is having proper blood pressure with the medicine, it will not go down that much, okay? So body is having a system to maintain it. And another thing that we are trying to use a dose, and we are trying to make it even better with telmisartan changing some side chain. Actually, I did not talk about that. We have already developed a few other molecules using telmisartan as a core molecule and adding other things onto it to make it better, so that we can even reduce the dose of telmisartan which can be given to the people.
Thank you, Madam. Very clarity, clarity. I have been very confused. Regularly, people are taking components, people are taking low body, that is, anything that they are not taking, that they are doing,
Yes, this is a very interesting question, and I am yet to do two experiments. One is to take a statistics. Actually, we have a BIRAC funded project where we are having 6 centers in all over the India, where we take the patient samples. And I am yet to see the data set where if we have some people who are taking telmisartan regular, whether we are seeing reduced viral infection into that. So these data sets will help us to know whether the taking of telmisartan, those who are taking, whether they are, kind of little protected from viral infection. And in the lab, what we can do is we can take the PBMC of those people who are already taking telmisartan, like maybe continuously, because BP medicine they will take every day. So if we do an experiment with that PBMC and then see they are kind of resistant or inhibitory to viral infection, I will clearly able to tell you the experimental value for it. But to my understanding, yes, because it is not only chikungunya. There are dengue, Japanese encephalitis. This work has been done, but it has been shown that 81 blocker is actually helping reducing viral infection.
Regarding the anti-viral and so what will be your opinion on that? Is it required? If it is required, randomly all the antibiotics is required or any kind of antibiotic is required?
Yes. So, we have very little antiviral drugs, as I showed. There are really more numbers to come. So antivirals, if you think about the viral fever, we have no idea what those viral fevers are, except like dengue one can taste, chikungunya also one can taste, JV one can taste. But the influenza kind of viral fever happens. 5 days there are fever and all. So their doctor is not even bothered what that virus is, right? Because we don't have any specific tip. What I will do by knowing also. So this is something that we should address. What are those viruses? So that we know the burden of the disease, burden of those particular viral disease, so that we can think of detection technique. We can think of a particular drug to control that. As of now, we don't have it. So what doctors do, they give antiviral. Why? Because as I mentioned, that when our immune system is down, then we are prone to other infections. The other infection can be antibacterial, antifungal. So doctor prescribed antibacterial, basically antibiotics. I don't think it is always necessary, but if it is persisting, if it is child or it is elderly or with other comorbidities where you are more prone to any bacterial infection, there maybe it can be given. But otherwise, one should wait, and basically you need to boost up your immune system. So you have to sleep properly, take food properly, drink more liquid. So my suggestion is that you keep your immune cells healthy so that they can fight better instead of taking antibiotics. No.
But is that and all these certain so many platforms are there. how we have technical sources, what is the result between this technical and legal activity. All this is also and also background derivatives are there. What is the structure behind this?
Yes, I have actually skipped that slide where all the 81 blockers, it can be any name. 81 blockers are used for this reducing leaking, and 81 is actually required for the viral infection. As I was mentioning, that virus does not have all the machineries for its own replication. So there are a lot of host factors which the virus is taking care of. And 81 and PPAR gamma. So telmisartan is a 81 blocker antagonist and PPAR agonist. So this is the mechanism what is required for the virus to replicate. So that is why if we antagonize and if we use 81 blockers, all that. Yeah. No, there are a lot of drugs. We have taken telmisartan as a representative of 81 blocker and PPAR gamma agonist. It can be anything with that mechanism of 81 blocker can work. Viral drug using this I am not aware, but 81 blocker telmisartan has been checked in several viruses, but not like a drug as of now. This is what we want to basically propose. So this there was a grant where we have worked on this. Now we have taken it to the next level, where we are developing different conjugates with 81. So that we can even use, actually we have filed a patent for it, because telmisartan is a drug where you cannot file a patent. It's already there. So the other drug we have filed a patent, and this other one that telmisartan, that also we have the patent is granted. So there we have added other molecules so that the solubility of 81 blocker will also improve, so that we can use less amount of drug to control the virus.
Yes.
With particular plant I have taken. So there are a lot of plants actually. I have only picked few from here. Yeah. So if you can see that there are amla and there are long bean, one bean is there in northeast, and here some goat meat, apola. These are some plants which are shown in here, but we have actually taken a lot of plants, more than 100, which are known medicinal plants, and also they are used for different fever, reducing fever. That we have taken. Also, we have around 20 plants, bamboo plants, which are known for its medicinal property. And so we have screened all this for chikungunya, Japanese encephalitis, SARS-CoV-2, herpes virus, and many of these plants are showing good effect.
Thank you, ma'am. Thank you for this very good session. Thank you.
Sorry that I stand in between you and your lunch. I did not want to hold you. Okay. Thank you for your patience here.
Thank you, ma'am, for such informative session. Thank you, sir, for your helpful comments. Now, I would like to request Dr. Praveer Kumar Sinha Mahapatra, Principal, Institute of Pharmacy and Technology, Salipur, to felicitate Chairperson, to felicitate the Speaker of the Day, Dr. Soma Chattopadhyay. Moving on, I would request Dr. Mrityunjay Banerjee, Associate Professor, Institute of Pharmacy and Technology, Salipur, to felicitate the Honorable Chairperson, Dr. Santosh Kumar Bal. Thank you, sir. I take immense pleasure on the behalf of the organizing committee of today's National Conference 2023 in extending our sincere thanks to the guest of honor, Dr. Santosh Kumar Bal, and Speaker of the scientific session, Dr. Soma Chattopadhyay, who gave us such an informative talk. Your presence put a significant imprint to our heart. Thank you very much.
So dear delegates, now it is time for lunch. Already we are 30 minutes late. So anyway, we should resume here by 2:15. Now it is 1:30. After 45 minutes, we will resume for the second session, scientific session. So we have arranged our lunch at Animal House building. So please, all delegates are requested to move there and there. And next session, our next talk is by Dr. Krishnamurthy Bhatt, Professor and Vice Principal, Department of UE, Manipal University. Topic is for our systems, strategy, safety, solubility, and viability enhancement techniques. Thank you.